In the name of Allah, the most beneficent and the most merciful, Assalamu alaikum. How are you all, dear students? I welcome you in Pakistan International School Types virtual learning system for the session 2020. And this is biology class for grade first year. And today is lecture number 20. Dear students, we are going to start today's topic nucleic acid in chapter number 2 that is about biological molecules. In the previous topics, we have studied about carbohydrates, proteins, and lipids, detailed account of the classification, their structure, and their functions. Today, we are going to see nucleic acid. So let's get started. Yes, dear students, nucleic acid, as we know that they are present in DNA and RNA, very important molecules which are present in our body. So actually, nucleic acids were first reported or discovered in 1869 when a Swiss physician, Frederick um, Frederick Micher, when he was studying about the chemistry of cells, so he just taken or isolated some pus cells which are rich in white blood cells. So he saw some of the substances which are present in them. They are when he studied that, so he found that they were not lipids, carbohydrates, or proteins. So that was new for Frederick Micher, and he just gave them name nuclein. So nuclein afterwards, uh, because due to its origin, because it was isolated from the nucleus, so in 1920, uh, like its complete structure and basic chemical nature has been found out. That's why its name is given as nucleic acid, because it is having some acidic properties and they were discovered from the nucleus. That's why its name given as nucleic acid. So if we talk about structures of nucleic acids, so nucleic acids are of two very basic types. These are called ribonucleic acid and deoxyribonucleic acids. If we just see later on their structure, so they are linear and unbranched polymers. Polymers of what we will talk about in detail, that how its units, they will just make do they will do polymerization by condensation, and what type of monomer they are having. Actually, deoxyribonucleic acid and uh, ribonucleic acid is having just first of all, very little difference, because ribonucleic acid is having OH group at carbon number 2, and here deoxyribonucleic acid is having no oxygen. It is having just hydrogen at the place. The monomer of nucleic acids is nucleotides. So if we go deep into the nucleotides, then we can say that deoxyribonucleic acid, that is named as DNA, and ribonucleic acid that named as RNA, that is having three basic units. What are three basic units? That number one is pentose sugar, number two phosphoric acid, and number three is having bases. So pentose sugar is the same for both of them, but very little difference is there. As I have told you, that in DNA, deoxyribose sugar is present, and in RNA, ribose sugar is present. If we see the structure of, first of all, DNA and RNA, both of them. So just we can see here actually this one. If you see this structure, this is the basic structure of a nucleotide, which is the monomer for RNA or DNA. Number one is pentose sugar. If you see this sugar, which is pentose, pentose means a 5-carbon ring is present here. The sugar which is present, that is having a 5-carbon ring. We have studied this in carbohydrates. So this sugar, if you we talk about RNA, so it is having OH group at carbon number 2, and if we talk about DNA, it is having um, H group at number 2. And let's talk about another group that is called phosphate group, so that is phosphoric uh, acid attached with that one. It is PO4, and it is having slightly negative charge, and that makes it acidic. That's why it is called as nucleic acid. Then we'll see the third part of this one, that is nitrogenous base, and this nitrogenous base are of various types. Because of unshared pair of electrons of nitrogen atoms which thus acquire a proton, you can see here nitrogen at this place, at alternate places. Nitrogen are present. That's why they are called nitrogenous bases, and that is actually gives like acquire a proton, and it means um, it is having unshared pair of electrons. So that's why it gives rise to the base structure. That's why this is called nitrogenous base. So bases are of two types. These types are purine and pyrimidine. If you see here, this is the ribose sugar of deoxyribose, which is present in DNA, and ribose sugar, which is present in RNA. Number 2 group is phosphoric acid. If you see the structure, this one. And next one is pyrimidine bases, and then purine bases. Purine bases are double ringed structure. One is 6-carbon and other is 5-carbon ring. And the other one is a 5-carbon, we can see, because nitrogen is present here, a 5-corner ring. So pyrimidine bases, they are having single ringed structure, and due to the difference of only one nucleotide, like one group here, it can be chained into different uh, pyrimidine bases. So we can see here pyrimidine bases, that is cytosine, uracil, and thiamine. So cytosine and thiamine, they are present in DNA, but uracil is replaced in the like in place of thiamine in RNA. So in place of thiamine, we are having uracil in RNA. So if we talk about purine bases, so you can see here there are two purine bases, adenine and guanine. Adenine and guanine, they are double ringed structure, and you can see they are having the differences here as amino group here, and it is placed here at carbon number 2, and it is placed here at carbon number 6. Okay, so now we will see that how the bonds between these all units are going to be formed. So while during formation, first of all, nitrogenous base is linked with pentose sugar, because the pentose sugar and a phosphate group, they make the backbone of RNA or DNA. How they will form the backbone of RNA or DNA, we will see in a few minutes. So we can see the first group, that is pentose sugar. Here at carbon number 1, nitrogenous base is combined with uh, this carbon number 1 at pentose sugar. These two portions in which phosphate sugar, pentose sugar and this any nitrogenous base which is going to combine with this one, the both units collectively known as a nucleoside. Later on, one phosphoric acid is joined with pentose sugar at carbon number 5, not 4. Carbon number 5 of pentose sugar is up here out of the ring. So this phosphoric acid is attached with carbon number 5. So now this nucleoside plus phosphoric acid, that is called nucleoside monophosphate. If there are two phosphate groups, nucleoside diphosphate. If there are three phosphate groups, so they are called nucleosides triphosphate. So this is called as one nucleotide. Nucleoside monophosphate or nucleoside diphosphate or nucleoside triphosphate, they are called nucleotides. So before going to make DNA and RNA, there must be three phosphate groups. So what will happen? This is the structure as we have studied in ATP. We have studied this in ATP, that here at the place of this nitrogenous base, adenine is attached here, and here three phosphate groups are attached. That's why it's called ATP, adenosine triphosphate. So but when two phosphate groups are detached from that, that is called pyrophosphate. When this pyrophosphate means when two phosphoric acids are detached from that, so they are called, what they are called, you can see here. This is the structure which is going to make like this. In this, one more nucleoside triphosphate is attached. So what happened, the pyrophosphate, these two phosphate groups collectively are called pyrophosphate. They are detached from here, and uh, this phosphate will make the bond with this three carbon, in which there is hydroxyl group. So hydrogen will be removed, and then it will be uh, this phosphate will be attached with this one. So here what will happen, phosphodiester bonds will be formed. You can see here that this phosphate, it attached from one side with hydroxyl group like here, and at the other side it is attached with carbon number 5 at pentose sugar. So this is 5 to 3, and like at one side it is attached with 5 carbon, and it is other one it is attached with 3 carbon. Like the 5 phosphate will be attached with 3 carbon of this pentose sugar from its 5 end side. So this is called 5 to 3 end side, and here is a small comma is there, that is called prime. So 5 and attaches to 3 end That's why it is called 5 end of this pentose sugar will attach with 3 end of this one. That's why it's called 5 to 3 prime end. So and this polymerization of this like long DNA and RNA chains, they are formed in 5 to 3 prime end directions. So what is going to happen? Phosphodiester linkage will be formed, and then remove a phosphate group. Unlike proteins, carbohydrates, and lipids, where by condensation a water is going to be removed, but here pyrophosphate group is going to be removed. So how these nucleotides are formed, when pentose sugar and nitrogenous base is attached, that is called ribose, and a base is attached. If adenine base is attached, that is called adenosine. If guanine is attached, that is called guanosine. If cytosine is attached, that is called cytidine. And if thymine or uracil is attached, that is called uridine. Actually, that is in RNA. So we are talking about in RNA because uracil replaces a thiamine in RNA. So how the nucleotides are formed, that ribosomes plus base as well as a phosphate group is attached. These three groups when attached, so they will call adenosine monophosphate, then adenosine diphosphate, then adenosine triphosphate. First triphosphate, so when there is the nuclear long chain of polymerization is to be done, so there is two phosphate groups are DH, that is called pyrophosphate. Then if one phosphate is attached, that's called GMP, like guanosine monophosphate, guanosine diphosphate, and guanosine triphosphate, or that is called nucleotides which are present in ribonucleic acid. So this can be happen up to the uracil. All four nitrogenous bases are attached in this manner. So in DNA, the same process is done, but we just add a D because that is related with DNA, like deoxyribose. When attached with the base, so it is called adenosine, then guanosine, cytidine, and a thymidine, and then if we just gonna make a deoxyribonucleotide, so it will be having D with that one, and all the formation is like the RNA except a thymidine, which is replaced by uracil or uridine in RNA. So these nucleotides, they are polymerized and formed polynucleotide long chains of monomers. So later on, which formed DNA. So we are having certain other nucleotides except DNA and RNA. They are called ATP, like nucleotides which we have talked about before. And we are having NAD also. What is ATP? Adenosine triphosphate, that is mononucleotide, as we have seen in the table before, and they are connected with a covalent bond with one another, like adenine, nitrogenous base, that is nitrogenous base, ribose, pentose sugar, um, as well as three phosphates are attached with that, and two covalent bonds linking three phosphates together. If you see the structure here, so this is adenine, a triphosphate. These two make adenosine, then adenosine monophosphate, adenosine diphosphate, then adenosine triphosphate. This molecule is formed like this. Okay, we know that ATP is energy currency, as we have studied in the previous classes as well, and there is ATP is made from the oxidation of organic molecules during respiration. So the energy process, like ATP formation process, is respiration, cellular respiration, which we have seen in the previous classes in grade 9. And energy is formed when what will happen, this one bond of phosphate is broken down uh, and it will form ADP, and then one more bond will break down, so then it will form like AMP. So what will happen when this phosphate group will detach from that, and this bond will be broken down, so it will release 7.3 kilocalories per mole energy. So this per mole energy is utilized for our like different body functions. So that is happen in oxidative phosphorylation. So when we attach, like in respiration, energy is going to be formed. So when attach ADP with one more phosphate group, so what happened? This whole energy is stored in that, and then we can utilize this energy later on after breaking this phosphate group in many processes. So we will see in chapter number 4 in detail about oxidative phosphorylation that is happened in mitochondria. If we talk about nicotine amide adenine dinucleotide, the name is telling you here nicotine amide and adenine. These are two nucleotides are attached with one another with so this is called a dinucleotide because it is containing nicotine amide adenine adenine, both of the nucleotides we can see here in the structure. You can see this is one nucleotide and this is other nucleotide. So they are attached with one another with phosphate bond, with phosphate linkage. Okay, so that's why you're forming a dinucleotide, and that's why they are called nicotine amide adenine dinucleotide, and that it acts as like coenzyme in lots of processes, and it acts as oxidizing agent with hydrogenases, like where we remove hydrogen. You can see here, so there is a reduction process, like hydrogen is going to be removed here, so it will, or if it is sorry, when it is hydrogen is added, so that is called NADH, which is high energy molecule, and when um, hydrogen will be removed, so that is the oxidized form NAD, which is utilized as a coenzyme. Okay, so let's talk about a very important topic regarding DNA and RNA structures. Dear students, in 1951, what happened? Erwin Chargaff found that nitrogenous bases in the DNA show specific ratios, and he found that all the DNA, all the nitrogenous bases which are like present in DNA, they are in equal amount. Like if thymine is 1, so guanine will also be 1. Okay, here one thing more is going to be pointed out, that we have seen nitrogenous bases, so they are complementary with one another. All adenine will bind with thymine always. Always adenine will bind with thymine, and if we just talk about, they are bound with double bond, and then guanine will bind with cytosine with a triple bond. They are always complementary with one another. They can't be changed. It cannot be happen that T join with A and A join with T, but here at the place of thymine, uracil is replaced in RNA. So pyrimidine with bind um, uh, with like that is called purine, and purine will bind with pyrimidine bases. So they are present in one ratio 1. As much guanine is present, as much cytosine will be present over there. So this conclusion is known as Chargaff's rule. So after this description, X-ray diffraction analysis also done on DNA, and Maurice Wilkins and Rosalind Franklin public this all study of X-ray diffraction analysis of DNA, and they first claimed that DNA is not a single helix, that is duplex or double helix molecule, means they are present of two polymer chains or two polymers of these nucleotides sequence, and they are having width of 2 nanometer, while the length of each turn, like they are twisted around, so they are having each turn 3.4 nanometer wide. So after that, in 1953, so James Watson and its colleague, that is Francis Crick, both of them has they were doing the work and research on the structure of DNA. So they found or they formed the DNA model that is called Watson and Crick model, which you can see here. They were just examining, or actually the in the formation of DNA model of, you can see here, it is twisted around, and in their lab they have made this model with the different instruments. So they were gonna describe its structure later on. You can see very clearly like this. Okay. So what happened in DNA model of DNA model of Watson and Crick? So he said that it is made up of two polynucleotide chains, as X-ray diffraction told the structure. So in this manner, one chain runs from a 5 to 3 prime end, and you can see here in this structure, this is, like you can see, yes, these are this is double helical structure of DNA, and you can see this is a 5 prime end and this is a 3 prime end. It means that the bonds which are going to form, they are at the 5 to 3 prime position, as we have discussed before. So you can see here the pentose sugar and all the nucleotides which are formed here, they are in upward direction. You can see this structure of pentose sugar O group that is present up on the direction, and to form another helical structure, because these nucleotides have to bind with one another, so it has to be twisted upward, downward, up to down. Okay, so when it turns up to down, then it can make bond with other nucleotides. So complementary of this or opposite of this, if this is 5 prime end, so this will be definitely 3 prime end, because OH group is present here at carbon number 3, and this phosphate group will be attached from here at carbon number 5. So this is 3 to 5 prime end and this is 5 to 3 prime end, so it will move upward like this and it will move downward. They go parallel with one another, and due to the like this hydrogen bonding between these nucleotides as well as among themselves, what happened, they they twist around one another. One more thing, if we see, this is And the one nucleotide to other, there is a difference of 0.34 nanometer, and from one to this whole twist it just takes 3.4 nanometer. So tri or thiamine will always bind with A, and G will always bind C, like guanine with cytosine with triple bond, and this one T with A with double bond. And one more thing is to be noted, that phosphate and pentose and phosphate sugar, this group, this will be always outside, and this will make the backbone, but this backbone is outside, because these nitrogenous bases will make the bonds between one another, as well as this portion of like this pentose sugar and phosphate group, this is having slightly negative charge, as well as it is hydrophilic in nature. That's why it moves outward. And this inside portion, this one where the nucleotides are present, it is hydrophobic, so it just keep itself in between. That's why the backbone of phosphate pentose sugar, that will be always outside. So we can see here the structure of DNA, that it is present. We know that it is a double helical structure, which is twisted around so many times and around the histone proteins, and it makes the chromosome. And you can see in the zoom in structure where the phosphate backbone is outside, and in between there is the nitrogenous bases. Here we can say one more, we can see one more structure of that. You can see how the polymerization of this is done. This is deoxyribose, like DNA structure. Here is the ribose structure. You can see here OH group. So you can see phosphate is attached from here at carbon number 3, and from here at carbon number 5. 1, 2, 3, 4, and here is 5. So this is attached from 5 then to 3. So this is 5 to 3 prime end, and this is 3 to 5 prime end. These are adenine and guanine purine bases, and pyrimidine bases are cytosine, thymine in DNA, and uracil in, we can see here uracil in RNA. So we can see the differences between RNA and DNA. DNA is double helical structure, but RNA is single helical structure, but that is can also be twisted around like this. But here it is like this. Okay, so here uracil is replaces the thymine group, which is present here. So actually RNA derived from DNA, which we'll talk about in the next topic. So here you can see its nucleotide units as well. So this is sugar phosphate backbone, which will be always outside of that molecule. Yes, dear students, I hope you understood this lecture. Very informative. Inshallah, we will see you in the next lecture, in which we will see about some more information about the genes and the DNA in our next topic. Allah Hafiz. Wish you all the best.